We have come to the second learning objective of this lecture, which is phloem and translocation. Phloem and translocation involve the movement of organic solutes, for example, sucrose, amino acids, organic acids, potassium, chloride, phosphate, magnesium, from leaf of we know before as a source or site to produce or store the food from the process of photosynthesis to sieve tube in phloem to be carried out to other parts of plant or known as sink or a place or region or site which need the food for storing, for living. Unlike the movement of xylem sap, the movement of phloem sap requires energy expenditure on the parts of the plant. This is certainly because in xylem sap, it only involves the transportation of water and dissolved mineral, which is less concentrate, and also small molecule size. However, in phloem sap, they are high concentrate and bigger molecule size. Therefore, the transportation of phloem sap requires energy expenditure. I will explain to you how solutes move around the phloem according to the pressure flow model. This model of pressure flow to show the mechanism of how solutes move around the phloem can be explained by consideration of osmotic motion applied to solution of two sugar solution across a semi-permeable membrane. Why they said this pressure flow model can be explained by consideration of osmosis motion applied to solution of two sugar solution across a semi-permeable membrane. This is certainly because the sucrose from source need to be transported into the xylem vessel, which they need or required to cross the semi-permeable membrane not only of the source cell but also the phloem vessel. Carbohydrates, or to be specific, the sucrose, are actively transported into phloem at source. This high concentration of carbohydrate will cause greater osmotic pressure in phloem. And because of this condition of greater osmotic pressure caused by high concentration of carbohydrate in phloem vessel, it will cause the water to move in from adjacent xylem by osmosis. This is because high concentration of carbohydrate in xylem vessel will invite the water to move in following the concentration gradient. And this situation further will create water influx and therefore resulted with the pressure inside the Phloem vessel. And this water influx caused by movement of water from Phloem, creating the pressure inside Phloem, and finally this pressure are able to push water and dissolve carbohydrate through Phloem from one vessel to another vessel, or from one tube to another tube, until they reach to the destination of sink. At sink, the carbohydrate would actively remove or unloaded from Phloem vessel into the sink. This will create or reducing the osmotic pressure in Phloem. This is because the high concentration of sucrose or carbohydrate is now no longer there because already being transported or unloaded from the Phloem vessel into the sink. Left the water in the Phloem vessel. And because of this situation, the water will leave the Phloem and re-enter the xylem. And finally, they will maintain an osmotic pressure gradient between the source and the sink. This is another diagrammatic features of law to show you the pressure flow model in Phloem and translocation. We will start with the sugar which were manufactured in the leaf or a source will diffuse to Phloem companion cell. We know that companion cell, they are located in between the source and the Phloem vessel. They function as a temporary storage area before they need to be unloaded into the Phloem vessel whenever the requirements are. The sap, which is sugar, dissolves in water at high concentration. Since this is high concentration solutes, the plant must use active transport to work against a diffusion gradient as part of the sap moving process. This is certainly because the biological theory of solute transport, whenever the solutes need to be transported against the concentration gradient, therefore they require energy because they are now moving against a diffusion gradient. The companion cells will actively transport the sap into sieve tube of the Phloem. And here they come, the water from the xylem vessel following the osmosis from source cells and xylem. And this creates high pressure. The pressure is because the combination of mass flow of water from xylem together with high concentration of sucrose in the phloem vessel will result with the push and pressure of sucrose from one phloem vessel to another until they reach to the sink. At the sink, this high concentrated sugar will diffuse out of the phloem and followed by water by osmosis. And this occurrence will create low pressure because there is no longer retaining of high concentrated of sucrose. There is no longer high concentrated or high mass of water in the phloem vessel. Sugar solutions flows from high to low pressure right from the source until the sink. And at the end of the day, water may be taken up back by the xylem through the transpiration stream. This is the second recap of how the pressure flow model in phloem and translocation process takes place. Let's look at it again. We have sucrose produced by source need to be transported into the phloem. And before that, these sucrose have to pass through the companion cell. And before they pass through the companion cell, this sucrose requires energy because they are moving or crossing a semi-permeable membrane with high concentrated of molecular mass moving against the concentration gradient. Therefore, in this process, they require energy. Once the sucrose loaded into the phloem vessel, therefore, it creates the situation where the water will move in, following the potential gradient by osmosis. Because of the water influx from xylem into the phloem vessel, it will create a further pressure, and finally the pressure are now able to push the content of sucrose into the next following phloem vessel until they reach to the sink. Once the high pressure of sucrose together with the water reach to the sink area where they are required to unload it in, this sucrose will be transported into the companion cell and finally to the sink. And this process creates low pressure potential because there's no longer high concentrated of sugar together with the water, which initially created the pressure to move the high concentrated of sugar from one vessel to another. This low pressure potential now permitted the water to re-enter or return into the xylem following the osmosis. In this slide, I am sharing with you the circle or chain of pressure flow model, but in written. You can have a look at it and doing your own reading, especially to further understand about how the pressure flow model involved in Phloem and translocation. Talking about the Phloem in conducting translocation according to the pressure flow model, what makes the Phloem so special, so optimum, until they can do the work continuously to support the sustainable, to support the life cycle of the plant? There must be a certain features or a certain character that the Phloem vessel have. In this lecture, I would like to share with you there are two key characteristics of Phloem vessel which make the Phloem optimally function to transport food throughout the plant structure. The first character is the sieve tube, followed by companion cell. In sieve tube, they are long cylindrical structure consisting narrow living elongated sieve element which join together. And at the end of each sieve element or Phloem vessel, the end wall meets to form sieve plate and plasmodesmata which enlarge to form the sieve tube. We know that by having long, cylindrical, narrow, and elongated sieve element which join together ideally will help the Phloem to transport the food effectively. And by having plasmodesmata which enlarge to form the sieve tube, it will help the transportation of a certain molecule size to be transported from one Phloem vessel to another. When mature, nucleus, Golgi apparatus, and ribosome degenerates in Phloem cell. And a thin layer of cytoplasm and some small mitochondria which found lining inside of the thin cellulose cell wall. This presents less barrier to flow of cell through sieve element. This representation of a thin layer of cytoplasm and some small mitochondria which found lining inside of the cellulose cell wall really helps to present less barrier to flow of cell through the sieve element. What happened when this sieve tube rupture? There must be a certain problem, difficulty, or malfunction if the Phloem vessel, especially the sieve tube, rupture. So how to mitigate? So how plants mitigate this situation to reduce the malfunction of it? Therefore, in nature, when sieve tube rupture, the protein strand will form plug to block the pore. Where do these protein strands come from? We know that phloem transport amino acid. It is a monomer. It is a building block of protein. Then it will replace of the original sieve tube by joining and combining, becoming a polymer of protein, and they will strand to form a plug to block the pore. They will substitute, they will replace the function of a sieve tube when the sieve tube ruptures. Other than that, callus deposit, as in any form of waste transported by the phloem vessel, they will also take place to form across the sieve plate to prevent loss of phloem sap. And in nature, we can see in any difficulties, there will be the trigger to help it to restore the functions automatically. The second characteristic of phloem is companion cell, which is associated with sieve tube. However, they are smaller, shorter compared to sieve tube, which are long and elongated. Companion cells contain large nucleus, dense cytoplasm with numerous mitochondrial ribosomes and endoplasmic reticulum. These cells are metabolically very active, connected to sieve tube element by plasmodesmata. They need to be metabolically very active and connected to sieve tube by plasmodesmata because companion cells are located between the source cell to the phloem vessel or the sieve tube. Therefore, they need to be active in receiving the food and also distributing the food. Receiving the food from the source and distributing the food to the phloem vessel or the sieve tube. That's why they need to be very active and connected to sieve tube. By having plasmodesmata, it will further help the effectiveness of companion cell to receive and to distribute. The final slide of this section is to show you the diagrammatic features of phloem. As what we know before, as what I've mentioned before, as what I've explained before, phloem vessel or phloem are made up of a sieve element joined together with another sieve, and at the end of each of the sieve, there are sieve plate, function to filter, function to screen, function to select any required size of molecule to pass through from one element to another. And they are accompanied by a companion cell which are metabolically very active in order to transport the food from the source to the sieve element. They are further strengthened by fiber. With that, thank you very much to all of you. Thank you very much for your participation. Thank you very much for your cooperation in listening to this lecture session. I hope in some part it will help you to further understand, it will help you to better understand about plant, especially the physiological part of the plant, by understanding how the plant conduct the water transport and gases exchange within the whole structure. And I do hope it will help you to further understand the functions of the plant in this world. If you have any question, if you have any query regarding this topic, feel free to contact me personally through email, through phone, or you can even come to my office at any time that we are mutually agreed to meet. Once again, thank you very much, and I really hope in some part it will increase your understanding about plant science. Assalamualaikum warahmatullahi wabarakatuh, and salam bahagia.